Hello, everyone, and thank you for coming to the session. My name is Soha Kenj. I'm Professor of Medicine and Chair of the Infection Control Program at the American University of Beirut Medical Center in Lebanon, and I happen to be the President-elect for ISAC. And it's really my distinct pleasure to present the speaker for this session, Professor Sharon Lewin. Professor Lewin is the inaugural director of the Peter Doherty Institute for Infection and Immunity. This is a joint venture between the University of Melbourne and the Royal Melbourne Hospital. She's professor of infectious diseases at the University of Melbourne and a consultant infectious disease physician at the Alfred Hospital and Royal Melbourne Hospital. She's also a National Health and Medical Research Council practitioner fellow. And Dr. Lewin did her PhD at Monash University and then her postdoctoral fellowship with Dr. David Ho at the Rockefeller University. So again, it's my pleasure to welcome her to the podium. She will be talking to us on the topic from HIV to COVID-19 and beyond, the role of antivirals in responding to pandemics.
Thank you very much, Suha, for the kind introduction. And I think I'm standing between the audience and a swim in the 36-degree heat of Perth. So thank you for coming along in the late afternoon. I'm pretty sure everyone in this audience is well aware that the HIV pandemic has been totally transformed by the availability of antiviral therapy. This shows you the number of AIDS-related deaths that have continued to decline since the mid-90s globally with the increasing access to antiretrovirals. And now, antiretroviral therapy is available to 70% of the global population of people living with HIV and getting cheaper and easier to take. At the same time, antiviral therapy has had a remarkable impact on the incidence of new HIV infections, not what we normally think about for treatment. We first learnt this was possible by the description that treatment is prevention, meaning that people living with HIV on antiretroviral therapy are unable to transmit HIV to their sexual partners, what we commonly call untransmissible, undetectable equals untransmissible. And at the same time, availability of oral antiretroviral therapy and now injectable antiretroviral therapy incredibly protects an individual from acquiring HIV infection. So even without an HIV vaccine, we are seeing significant declines globally of HIV incidence in cities with high uptake of ART and PrEP, cities such as London, San Francisco, Sydney and Melbourne, and antivirals on their own are reducing the incidence of HIV infection, which is quite extraordinary for an infectious disease. Having said that, we still have 1.8 million new HIV infections in 2020, and really we have a major challenge in implementation. And that's one of my first points I'd like to make. No matter how good the science is around vaccines, antivirals, diagnostics, we always have this challenge of implementation. Now, we've learnt that with COVID-19, there are also multiple potential roles of antivirals. Antivirals can reduce the individual's risk of acquiring infection, can reduce the risk of acquiring disease, and reduce the risk of requiring hospitalisation. We know that antibodies are able to do all three, and oral antivirals at least reduce the risk of acquiring disease and hospitalisation. So today, over the course of this talk, I'm going to first review what's available for direct-acting antivirals for COVID-19. I'm going to talk a bit about why antiviral development was so delayed and quite difficult actually for COVID-19, what we might do differently in the future using a totally different approach to antiviral treatments, an example of these being RNA targeting therapeutics that my own group have been working on quite extensively over the last 2 years, and reflect a bit about what might be needed for the future when thinking about pandemic preparedness. So current direct-acting antivirals for COVID-19, I'm going to review what's available largely and recommended in Australia. So this graph or picture I'm sure is very familiar to many people in the audience, beautifully illustrating the natural history of COVID-19 with transmission from asymptomatic to symptomatic infection, mild, moderate, severe and critical illness. And it's a great schematic of understanding the pathogenesis of COVID-19, but also the strategies for treatment. Now in Australia, we've had a fantastic initiative that started during COVID, is now continuing in other infectious diseases, the Australian COVID Evidence Task Force recommendations, which has been hugely helpful to all of the clinicians battling the barrage of new evidence we all had to face with COVID. And the recommendations are divided into recommended, conditional recommendation, consensus recommendation, and only in research. And currently, this is the recommendations for treatment of COVID-19 in Australia. You can see that there's a range of strength of these recommendations. And I'm really only going to talk to you about the role of antivirals early in the natural history of COVID-19 infection. And I'm going to talk to you about the TGA, or the Therapeutic Goods Administration, which is our regulatory authority in Australia, and what their recommendations are for early symptomatic treatment. And I acknowledge actually that these drugs are not widely available. This is nothing like HIV. Very few low and middle income countries have access to direct-acting antivirals. Just to briefly remind you the mechanism of action of oral antivirals for COVID-19, which were largely designed using similar approaches to oral antiretrovirals. This just shows you the schematic of the life cycle of COVID-19, starting on the left, attachment and entry of the virus, translation of viral proteins, proteolysis, RNA replication, transcription and translation, and then assembly, packaging and release. So the targets of antiviral activity are, of course, at viral entry using antibodies, the viral protease using Paxlovid, and RNA-dependent RNA polymerase, which is the target of molnupiravir and remdesivir. So Paxlovid, I'm sure you're all aware of the original licensing study. Just to remind you, it was a double-blind, randomised control trial in symptomatic, unvaccinated, non-hospitalised adults at high risk for progression to severe COVID. Probably one of the last studies designed like this. These studies would be impossible to do now in a vaccinated cohort. People were randomised to receive 5 days of Paxlovid or placebo, and importantly, the inclusion criteria were non-hospitalised patients with risk factors for progression, either age, BMI, diabetes or other comorbidities, and a fairly large study of around 2,000. I'm sure everyone knows these data, and clearly a beneficial effect of Paxlovid in reducing hospitalisation or death from any cause by day 28, with a remarkable reduction of 89%. So the next questions have been, does this translate into the real world? What is the clinical activity in vaccinated people? What's the benefit of antivirals in younger people with no comorbidities? And are we going to see similar efficacy in the real world? Well, there have been a number of these studies being performed and reported, initially from Hong Kong and then Canada, but I like the Israeli study best because of its size and design. This was a retrospective case review of people greater than the age of 40, so antiviral treatment wasn't restricted in the same way as it is here in Australia. Over 100,000 people, looking at the association of Paxlovid with hospitalisation and death due to COVID, and adjusting for important socio-demographic factors, coexisting conditions, and previous COVID immunity status. And this is what they found. Basically identical findings in the participants that were over the age of 65 years. You can see that on the left, a remarkable reduction in risk of hospitalisation in people aged over 65. And on the right, between the ages of 40 to 64, you can actually see no real benefit. So these countries, there are many countries in the world, have free access to antiviral utilisation. Really depends on what you're treating here, but if you're doing it to protect your health system and hospitalisation, it really only works in the elderly, greater than 65, or other comorbidities. And interesting, the Israeli study also showed us that a reduction in hospitalisation in people over the age of 65 was still similar in unvaccinated and vaccinated participants. Your absolute risk is lower, of course, in vaccinated participants, but you had a similar level of reduction. So really encouraging, and similar findings actually in the study specifically from Hong Kong and Canada. Then there's the issue of Paxlovid rebound, and is it real and what we need to do about it. This is a schematic of 3 cases in a case series reported in the New England Journal of Medicine, and showing you in red the days at which the individual was antigen positive, showing you in blue when they took Paxlovid, and showing the frequency or tempo of rebound of antigen positivity with the recurrence in the red squares following administration of Paxlovid. So on average, rebound at days 8 to 10. And there were some initial very high-profile cases of Paxlovid rebound. We heard about President Biden, we heard about Tony Fauci. Suha and I were just talking about David Ho, where David Ho is the second individual on this schematic. I'm sure he wouldn't mind me mentioning. That's the 69-year-old man who was visiting France when he got COVID. And even Peter Doherty had Paxlovid rebound. So it seems to us that this is pretty common. We keep on hearing about it all the time. And it's unclear, really, it has been unclear how common this really is. First, and most importantly, rebound is drug-sensitive virus, so that's very encouraging. Paxlovid resistance has been developed in vitro, but really not yet described in vivo. And there now are some large retrospective case series studies looking at how common this is. This is one study that looked at over 13,000 adults actually treated with either Paxlovid or molnupiravir within 5 days of their COVID infection. And they found that rebound post-Paxlovid at 7 days was 3% and at 30 days was 5%. Similar levels with molnupiravir. Rebound was more common in people with comorbidities and also those who have had a history of vaccination. So the pathogenesis of this is very interesting. It's probably not that common. The original studies of Paxlovid in a randomised placebo-controlled trial showed that the incidence of rebound in those that received placebo was about 1%. So it's probably slightly higher in vaccinated communities, but it really isn't a reason not to take Paxlovid. What do we do with Paxlovid rebound? We don't know. There's been no studies of what the best treatment is. The current recommendation is probably not to treat, but further studies are really needed to understand the benefit of retreatment or longer treatment. But actually, it's surprisingly rare. A really interesting report recently on the benefits of antivirals for COVID is its potential effect on post-acute sequelae of COVID-19, or another term for what's commonly called long COVID. And this study was done at the US Department of Veteran Affairs by a group that's done a lot of work on long COVID, given their capabilities in large data management. And the study looked at people that were tested and found to be SARS-CoV-2 positive between March and June of 2022, were not hospitalised on the day of the positive test, had at least one risk factor for progression to severe COVID-19 and survived the first 30 days after that diagnosis. And the study compared those treated with oral nirmatrelvir or Paxlovid within 5 days after the positive test, about 9,000 individuals, to those who received no antiviral or antibody treatment. And the control group had about 50,000 participants. And this is very interesting. The incidence of post-acute sequelae of COVID-19 was really pretty significantly reduced, a reduction of 74%. This was a small absolute risk reduction because post-acute sequelae of COVID is pretty rare, but an absolute risk reduction of 2% per 100 persons at 90 days. So very, very interesting to me. Does that mean that more people need to be treated for COVID in the early days? We don't know the answer to that. Does this mean that persistent virus is responsible to long COVID sequelae? I suspect not, because this was treatment at the very early time of infection. I think it's telling us that the burden, the viral burden that you're exposed to in that first week of infection determines long-term outcomes. Teasing out who should be treated to prevent long COVID, what the real cost effectiveness of this intervention and the number needed to treat is going to be challenging, but a very interesting insight into some of the drivers for post-acute sequelae of COVID-19. I'm going to switch briefly now to molnupiravir, also the licensing study that I'm sure you all know, a phase 3 double-blind randomised placebo-controlled trial that evaluated molnupiravir, again starting within 5 days of the onset of signs or symptoms, non-hospitalised, unvaccinated individuals, mild to moderate disease, and at least one risk factor for severe COVID-19. Not as effective as Paxlovid, and an estimated reduction in likelihood of infection of 30%, a hazard ratio of 0.69. Now, molnupiravir has also been assessed in younger and less sick patients. This is an interesting study that's come out of the UK called Panoramic, which was a different design. This was an adaptive multi-arm platform randomised trial. It had different inclusion criteria. It included people aged over the age of 50, so a lower age cutoff, or over the age of 18 with comorbidities. They'd been unwell for less than 5 days, confirmed COVID in the community, and they looked at the impact of molnupiravir relative to usual care. And this is time to symptom recovery. So there was a reduction in time to symptom recovery. This is just the preliminary analysis. This is still in preprint form. Large study, more than 25,000 participants. Interestingly, no difference in hospitalisation. This is why we use antivirals. But hospitalisation rates were very low at 0.8%. Time to recovery was reduced, may well be important for our workforce, reduced to 9 days with molnupiravir compared to 15 days with usual care. And interestingly, SARS-CoV-2 virus in nasal swabs was below detection levels in 21% of the molnupiravir group compared to 3% in the usual care group, and they had lower viral load, which actually may be well relevant to transmission dynamics. Again, how we use these drugs at a population level is really determined by cost of the drug, adverse events, and therefore whether it's cost effective to use this to reduce transmission, I think is unlikely. Finally, remdesivir, just for completeness, because I don't think we're ever going to see the scale of use of remdesivir as we have with the oral drugs. This was a study done quite early, 3 days of intravenous remdesivir in outpatients with symptom onset within 7 days, so a longer window than the oral antivirals, more than one risk factor for progression, small study of about 562, and you can see there really was a benefit, so not a bad option for people that can't take oral remdesivir, oral Paxlovid or molnupiravir for adverse events or contraindications. So we have a bit of a choice at the moment for using oral antivirals, and just like with HIV, our big challenge is implementation, awareness about them, making it really easy to people to access, and that's still, I think, a big challenge in this country and I'm sure in many others. What about antibodies as antiviral agents? We developed antibodies much, much quicker than we did for oral antivirals. Monoclonal antibodies can actually be synthesised very quickly from people with an acute infectious disease. I don't know whether many of you know how those antibodies are synthesised, but in brief, you can take blood from a person with acute COVID-19 infection or any sorts of acute viral infection, actually isolate spike-specific B cells This allows you to then measure and sequence the RNA in those individual antibody-positive B cells. Then you're able to actually generate these neutralising antibodies ex vivo and test them against different strains of virus. So this was done very, very quickly. The first antibodies we had were in June 2020. And the first antibodies we had, it was a Lilly antibody that's over on the left, and this is just a neutralisation curve. So you have a different concentration of the antibody on the x-axis and the percentage neutralisation on the y-axis, exactly like a drug IC50 curve. And you can see very clearly that these antibodies, which were the ones from Lilly and Regeneron, work beautifully against the early strains, Wuhan and Alpha, and by the beta strain we could already see that these antibodies were knocked out. And antibodies are complicated to administer. Initially, these were all given intravenously. We can at least now give them subcutaneously and intramuscularly, which it greatly improves their availability. But really, there are few or potentially no antibodies remaining active against new Omicron variants. And whether we will ever really be able to use antibodies as effective antivirals for COVID, I'm not so optimistic. Antibodies have, however, been the only intervention that's been demonstrated to help as prevention. So that's why I'd like to just briefly summarise this trial of Evusheld, which is a combination of two antibodies for prevention of COVID in the Provent trial. It's a phase 3 randomised controlled trial of Evusheld intramuscularly in unvaccinated participants. It was pre-Omicron. A very small proportion of these participants were immunocompromised, and it was unblinded early, so 40% unblinded as vaccines became available, so a short follow-up period. And you can see here that the time to first positive diagnosis was certainly reduced with receiving a single intramuscular injection of Evusheld. There was a low incidence of infection in both arms, 0.2% in Evusheld, 1% in placebo, but a relative risk reduction of 76%. So proof of principle that these, of course, work. The challenge is now what we do with emerging strains of virus that are resistant to Evusheld. The current recommendations in Australia are still to continue to use Evusheld for immunocompromised people at the standard dose. So what do we know about the new variants? Well, pretty much all current Omicron variants have reduced sensitivity to our current clinically available antibodies. This is a very nice summary in a preprint that shows you graphically the emergence of different Omicron variants in different parts of the world. The legend on the right is a little hard to read, but in blue is actually BA5, and you can see in countries like India and Singapore the emergence of several other strains that we're all quite worried about, XBB and BQ1 and BQ1.1. So how do these variants look when you test them against the range of either clinically available antibodies or experimental antibodies? Now, this is a very complicated slide, but what it shows you is the IC50s for a range of different variants. Each variant is a different row, and then each antibody is a different column. And the lighter the colour, the more resistant the antibody. So if the cell is white, it means that it's resistant to that antibody. Greater than 10 micrograms per mil is needed to neutralise that virus. And if it's dark red, it means it's sensitive. So you can sort of see there's lots of white in this summary of activity of antibodies against some of the new variants. The top row is D614G, the ancestral strain, effectively the Wuhan strain. And then the investigators looked at BA45, BQ1, BQ1.1, and down the bottom, XBB. And so if you just look at the antibodies that we're interested in, sotrovimab, which we were previously using a lot of, Evusheld on the far right, and bebtelovimab, which we don't have available in Australia, you can see there's quite a bit of white emerging there, and certainly these new variants XBB, BQ1, BQ1.1 are no longer sensitive to the antibodies. So antibodies, as exciting as they are, especially for me, because antibodies, this whole idea of developing neutralising antibodies to treat viruses actually emerged out of HIV. We don't have any licensed anti-HIV antibodies. The speed at which we got these was fantastic. They helped for a while, but they really have a limited shelf life. Now, on this diagram, you will have, there are some antibodies, of course, down the left-hand side that are experimental antibodies where it is still pink or red for these new variants. So there are other antibodies in the wings that will likely emerge, but whether this is really sustainable is unlikely. So I'm going to just sort of move now to a more philosophical part of the talk and a little bit more of research from my own group. So why was antiviral development delayed for COVID? We were all so proud of what happened with the vaccines, you know, incredible speed of development, vaccines within 12 months. Why couldn't we do that as quickly for antivirals? Well, I think there's a number of reasons why. First of all, the pathogenesis of COVID-19 is definitely complex. Those different phases that I showed you on the natural history clearly show an early phase of viral replication, a later phase of immunological disease, and it really did take a while to work out when to use antivirals, and we now know it needs to be used early. It wasn't obvious at the very beginning of COVID. Current available technology for antivirals actually takes time and can lead to some wrong turns depending on what cells you evaluate those antivirals in. We actually don't have any really good, highly active antivirals for respiratory pathogens. It's nothing like what we have for blood-borne viruses. And interestingly, there's been minimal investment in antivirals relative to vaccines for COVID. We did an analysis in October 2021 on the level of investment on new technologies in vaccines for COVID, which was estimated about $137 billion, and for antivirals about $7 billion. So we've spent about 5% of what we have spent on vaccines compared to what we have spent on antivirals. High throughput drug screen and rational drug design is how we design antivirals. This was basically how anti-HIV drugs were designed. So what do I mean by high throughput screening? It means taking the virus, sitting on top of a target cell and running multiple compounds through to see where you get the best hit or which compound may then inhibit viral replication. Then you then have a unique compound which needs to go through toxicity and evaluation, and it takes time. The other way is something called rational drug design, where you actually need the actual structure of your target. So this is an image of the HIV protease, and then you can design molecules to fit inside susceptible pockets of the protease. So to do this, you need to know the viral life cycle, you need to have isolated the particular enzyme and also develop the structure of that enzyme. So with COVID, we had multiple failed repurposed drugs. You know, hydroxychloroquine and ivermectin looked good in certain animal cell lines, but they didn't work in human cell lines. That's where we took a few wrong turns. And we landed with these 3 repurposed direct-acting antivirals, which I've already described to you. They're all actually repurposed drugs developed for influenza, Ebola, or SARS-1. So we still actually don't have any unique viruses that are highly specific for SARS-CoV-2. And this delay had great significance. So this is a rather complicated slide, but it shows on the top row the timeline for the development of the Pfizer vaccine with some key milestones with the pink triangles. And on the bottom line, the development of the Pfizer drug Paxlovid. And in brief, we had the Pfizer mRNA vaccine end of December and Paxlovid the following year. And again, we did some modelling to look at what would have happened if we'd had an effective antiviral drug just for people that got infected, not for prevention, and estimated we could have potentially averted about 4 million deaths, assuming that the drug is used and globally available. So what might be some new technologies for antiviral development that could allow us to move at much greater speed? Well, here's a schematic of the SARS-CoV-2 viral life cycle. In sort of yellow is the various targets, protease, RNA-dependent RNA polymerase and others. In the orange boxes are the different compounds that are being developed. But imagine if we didn't have to worry about understanding the life cycle. We didn't have to worry about understanding the actual structure of the viral protease and could take advantage of advances in gene editing. So you could just target the viral RNA itself, which would mean that on January the 10th, 2020, when we knew the sequence of coronavirus and everyone got to work on an mRNA vaccine, you could potentially get to work on an antiviral at those very early points in time. And this is, of course, now possible. This is Emmanuelle Charpentier and Jennifer Doudna, who won the Nobel Prize in Chemistry in 2020 for discovery of CRISPR-Cas9, that I'm sure you've all heard about, the ability to edit DNA with gene editing tools of CRISPR. But you can actually do this with RNA. There are different Cas enzymes that edit RNA. An example of those, one of those is CRISPR-Cas13. So very early in the pandemic, when everyone's labs were being closed, two scientists approached me. They work at the Victorian Comprehensive Cancer Centre. They're RNA experts and cancer experts. And on the left is Mohammed Fareh, and on the right is Professor Joe Trapani. And in about April 2020, they approached me and said, you know, our lab's closed down, we work on CRISPR-Cas13, and do you think this could be relevant as an antiviral drug? And I, at that stage, was receiving a lot of different requests on evaluating different drugs, and my first response was, what's CRISPR-Cas13? Because I actually never heard about it, even though I work on RNA viruses. They explained to me what it was, and I thought, wow, that sounds incredibly interesting and a very different approach to targeting viral replication. So Wei Zhao, who's a postdoctoral fellow in my lab, who'd previously been working on HIV and hepatitis B, got his hands dirty with SARS-CoV-2, and we began to look at whether this could be used as an antiviral treatment. And this is what CRISPR-Cas13 is. Cas13, you can see there, is the enzyme that cleaves RNA, and it's directed to the right spot by a guide RNA, which has sequence homology to your target nucleic acid. And you can see there that it's quite a long target, so it has very high specificity. You need a 30 nucleotide match in order for the enzyme to recognize and therefore cleave the viral RNA. So it's very easy to design because you just need the viral sequence. And that's what Mohammed did. He created a whole library of single nucleotide tiled guide RNAs that covered the entire genome. We didn't need to worry about which was protease, which was polymerase, etc., the entire genome. And then we evaluated whether those guide RNAs could do the job. And the first way we did it was using a reporter cell line. And I'll just explain to you what that is. So you can actually transfect in Cas13 itself and label it with the blue fluorescent protein, or BFP, the guide RNA itself, or the target. And the target here we used was a piece of spike RNA that we could manufacture, and it was tagged with GFP, or a green fluorescent protein. So you co-transfect all three things into the one cell, and if that Cas13 is active with the right guide, it will degrade your GFP. So you'll lose the GFP signal, or the green fluorescent protein signal, which you can see there on the right. And that's exactly what we saw when we used a non-targeting guide RNA, so an irrelevant RNA shown on the left. You can see there's no change in the GFP signal. And if you use a spike guide RNA, this is just one specific guide RNA 2, there's this dose-dependent loss of GFP. And we were able to show that with a range of guide RNAs in a range of different cell lines. We then moved on to look at whether this works with active virus replication, and here we essentially prepared Mohammed transfected the cells with Cas13 and the guide. He carried them over the road to the Doherty where Way infected them, and then he looked if viral replication was inhibited, and it clearly was inhibited with quite high potency. And again, we looked at a whole range of different guide RNAs that targeted different parts of the virus, because the spike is the worst bit to target because it's so highly variable. So really excited about this work, and the next challenge, of course, is delivering it in vivo. And how could you possibly deliver this in a very efficient way? Well, again, another great story from the pandemic, and that is that lipid nanoparticles and mRNA not only can deliver vaccine proteins, which each of you have received through your COVID vaccination, you can actually use mRNA lipid nanoparticles to deliver therapeutics, and that has been done. It's been done to deliver CRISPR-Cas9. It's reported clinical trial in the New England Journal last year looking at in vivo gene editing for amyloidosis using CRISPR-Cas9. So we think that we could potentially deliver this directly into the nasal mucosa. We may or may not need to functionalise those lipid nanoparticles, potentially with an anti-ACE2 antibody, so it could find the right target. And we've started doing some of this work, and it's very, very interesting. So we have looked at a whole lot of different lipid nanoparticles, which have different tropism, actually. The one that you've received from Pfizer or Moderna are licensed compositions of lipid nanoparticles, but each particle has 4 different lipids, so you can manipulate the lipids quite easily. And then we label these lipids with a dye, DID, and then we pack the lipid with a reporter construct, or mRNA. And then we've been giving these lipid nanoparticles intranasally, intratracheally, or intravenously, and then basically measuring where they go. And actually what we've found is fascinating. So this is just where the lipid nanoparticles end up because they're coated in a fluorescent dye. You can see on the left, there's a little bit in the tip of the head, which is the nose. This is when they're given intranasally. You can see they're in the lung and they're nowhere else in the body, so they really stay in the lung. Are they functional? Yes, they are. This is we were giving the mRNA to express a fluorescent protein called luciferase, and you can see the lung lights up, but liver, there's nothing else. So we're really excited about this as a potential way to deliver RNA targeting therapies. And other groups are doing similar sorts of work. This is one example from collaborators at Griffith University in Queensland using siRNA, which has been around for a much longer time than CRISPR-Cas13, and then other groups using similar approaches. So this is a very interesting way that you could develop antivirals potentially at speed. We can certainly do better with antibodies, and the big challenge and the big interest in both vaccines and antibodies is to develop pan-sarbecovirus antibodies. And pan-sarbecovirus largely means coronaviruses in the entire family of SARS viruses. And the first description of this came from Singapore, where a really interesting observation that people that had SARS-1 initially and then were vaccinated against SARS-CoV-2 made fabulous antibodies that reacted with every different type of coronavirus. And others have also found these antibodies in individuals. We don't know who in the room may have those antibodies, but some of you may well have pan-sarbecovirus antibodies. And this is just one example of one report of a pan-sarbecovirus antibody 2-36 on the left, where you can see it neutralizes all human, bat, pangolin viruses, doesn't do much to MERS because it's in a different family. And actually antibodies are used in many diseases. So we've previously evidence of antibodies working in Ebola, antibodies working in Nipah, and many other emerging infectious diseases. What we need, though, is that they're broad and that we can easily produce them and also reduce the cost. So what's needed for the future in this area? There's no doubt we need significant investment in new technologies. As transformational as HIV antivirals have been, this is technology from the early 90s that we're using. So we're here many years later. We need far better new technologies. And there is a lot of investment happening in this. So the NIH have recently committed $500 million, $500 million US dollars. It's a lot of Australian dollars now because our exchange rate's pretty terrible. But $500 million US dollars to specifically develop new antivirals. They're funding 9 antiviral drug discovery centres. They're focusing on this list of viruses of greatest concern for which we have few antivirals. The Gates Foundation, together with Open Philanthropy and Novo Nordisk, have also got a pandemic antiviral discovery program. They announced their initial call for paramyxoviruses. Both of these programs have short timeframes, 3 to 5 years, but a real increased effort and interest into developing antiviral agents. And then earlier this year, we had the amazing gift from Jeff Cumming. So Jeff Cumming is a Canadian philanthropist who now lives in Melbourne, and he recently gave a gift to the Doherty Institute of $250 million over 20 years to establish the Cumming Global Centre for Pandemic Therapeutics. We aim to leverage this gift significantly over the next 10 years. The Victorian government's also provided $75 million, and the primary goal of the centre will be long-term mission-based funding to researchers to develop these novel new platform technologies that are needed to develop therapeutics for pathogens of pandemic potential and at speed, along the lines of what CEPI did for vaccines. And many of you will, of course, know of CEPI, the Coalition for Epidemic Preparedness Innovation. It originally had a goal that a vaccine platform needed to demonstrate that you could develop a vaccine to a new virus in 16 weeks. They now have a new goal that their funded researchers will be able to develop a vaccine to disease X within just 100 days. So this is a very ambitious target. But even if we were able to develop vaccines quickly, we really do still need to solve the implementation problem. And a striking statistic I recently heard that only 25% of people in low-income countries have had 2 COVID vaccines. So as much technology as we have, even with speed, the implementation issue needs to be addressed. The G7 recently announced their 100-day mission, and actually they've included vaccines, therapeutics and diagnostics. So we need all 3 with speed to respond to a future pandemic. And finally, just returning to the HIV story, and that is that global access or benefiting from science takes more than just discovery. I started with this slide showing that now 70% of people living with HIV have access to antivirals and the death rate is decreasing. But this didn't just happen by drug companies developing new drugs. You know, this took years and years of activism by people living with HIV, partnerships between politicians, activists, scientists, and it took a really long time. So if you think about this, it took us 40 years to get to where we are, to 70% access. So we need to really address this issue around equity of any of the science that we develop for really effective pandemic preparedness. So in summary, antivirals really play a very important role in a pandemic response, in addition to vaccines. I'm not for a moment saying vaccines aren't important. For COVID, those advances were way too slow. There are reasons why, but we need to do better next time. I've shown you one example of a platform technology, CRISPR-Cas13, high specificity and potency, works in vitro, now need to work out how to deliver it in vivo. Another silver lining of COVID is this advance in mRNA LNP biology, which have a significant impact on therapeutics. And really, we do need substantial investment in new technologies for how we develop antivirals and also antibodies. They need to be active against whole families of viruses, and we need these adaptable platforms that can rapidly change in the event of a new pathogen. So I'd just like to acknowledge the various funders that supported the CRISPR-Cas13 work, and particularly Wei Zhao, Stan Can, Paula Cavall and Danielle Fong, who were intimately involved in doing some of this work, and a fantastic partnership with Mohammed Fareh, Joe Trapani and many others, and James McMahon, who helped me with some of the review of the currently available antivirals. And I'm happy to take any questions. Thanks very much.